this tiny creature. So yeah, I did some work with these frogs in the past while I was in Brazil. So I'm Brazilian, I'm from São Paulo, southeastern Brazil. And I'm passionate about field work, and I love the forest, the rainforests, and I have a little bit of experience that I want to share with you about these tiny frogs that live in the top of the mountains in the Atlantic forest. So that's why I'm going to talk to you, and I'm going to show you some of these results I have studying these frogs. So to start the talk, I'm going to start talking about the forest, right? So the Atlantic forest is amazing. So we have lots of habitats and microhabitats inside the Atlantic forest. And that's why I keep being so passionate about it. So if you observe in this picture, I'm going to show you lots of pictures that I have been taking during my field work there in Brazil. So I have lots of expeditions that I'm leading, and we expect to have more and more information to understand what's the history, the evolutionary history of amphibians that live there and what they can tell about the diversification of the forest in situ, in the place, right? So yeah, if we think about the huge structure of the forest in the Atlantic rainforest, we can see there's no empty spaces. So we can see bromeliads, we can see lots of trees, big trees, bushes, we can see lots of habitats that amphibians especially can use, right? And if we think about the wetlands and like the water, that amphibians are also like super into that, right? So they need water to reproduce. And they use streams, waterfalls, they use lots of moist habitats, including the bromeliads, so they can use the ponds, the little ponds that the leaf of the bromeliads keep during the warm days and during the rainfall. So they breed, usually to breed in these places also, not only in the regular habitats that we are used to see. So we can imagine being an amphibian in this huge forest and in this huge amount of different habitats that we can choose to live, right? So another thing is that the Atlantic rainforest is distributed across the coast of Brazil. So we have remnants of Atlantic forest from the northeast Brazil until the south. And then with this, we can expect a latitudinal gradient, which means we have different climates closer to the equator and more mild temperatures when we go down, when we go south. And we also have an elevational gradient from the sea level. We can see in this picture I took from here, it was me. So we can see the sea level and you can see the top of the mountain. These mountains reach around 300 meters, but the whole range of elevation in Atlantic forest can reach from 0 sea level up to 3,000 meters of elevation. So we have a huge, it's not a huge as we think about Andes, for example, but we have differences in climate and also in topographic conditions between the top of the mountains, the lowlands, and also between the warmer and the mild temperatures in different latitudes across the Atlantic forest. So this is super interesting because then we have a lot of diversity that is associated to these habitats. And we can mention, for example, for amphibians, we know Brazil is the richest place for anurans in the world. So we know that we have around 1,000 species just for anurans. We are not like Canada, that you deal with lots of caudata, with lots of salamanders. And Jay told me I'm working with the red-backed salamander right now here. And now we might think about like, what is what it's explaining, what can help us to understand this diversity, the diversity the Atlantic forest harbors. So we might think about this different kind of environment and microhabitats that we can have, considering this latitudinal and elevational gradients that I showed you before. So thinking about the amphibians, what we have is more than 600 species. I told you the whole country harbors around 1,000. So more than a half of these species are living in the Atlantic forest. And they also have more than 74 different reproductive modes. And this is related, most of the, related to their habits, like concerning the reproduction, so the breeding. They can be breeding differently using different habitats, such as you can see here in a bromelia leaf. And they can breed without passing by the larval stage, so they can trespass this stage. They can go more terrestrial than other anurans. And we have different strategies to hold the eggs, to take care of the eggs and the tadpoles or the hatchlings. So there's lots of different strategies that these species use. And this diversity is what called my attention when I started to study these organisms. So I'm really interested in knowing more about the direct developer species. So these are those species that goes straight to the adult form. So this means that they don't have the larval stage. So they leave the whole development inside the egg. And then when they hatch, they are just miniatures, such as their parents. So I'm super interested in this kind of development, and this is a cool venue for research, because we can, for example, understand how the amphibians develop strategies to be in the land, right? If you consider amphibians such as organisms that are fully dependent on water at some point of their lives, and they reach the terrestrial environment, so they might be developing some cool strategies such as this reproductive mode. And yeah, so the habitat diversity that we can see, it's related to ecological and this morphological specialization that I was talking about. And this lead us to endemism. So we have also lots of endemism, lots of endemic species occurring in the Atlantic forest in general, but also in the top of the mountains. So the problem is most of the Atlantic forests are devastated. So our estimations are around 10%. So there's numbers above or below this. So you can see in this figure 8.5, but the most recent are around 13% of remnants of the Atlantic forest. So in this map, the yellow lines you can see were the original extension of the forest in the coast of Brazil. Now you can understand the latitudinal gradient I was talking about. And if you pay attention in the map, we have this brown dry condition in the middle of Brazil that split the Amazon forest from the Atlantic forest. They are different forests, but they were connected in the past. So we believe there's some processes acting in diversification of flora and fauna that are occurring similarly in both biomes. But the main problem here is that we have devastated our forest in the coast, and then now we have to deal with this, like lots of endemism, a higher diversity, and also high levels of disturbance. So there's problems with habitat loss. So we don't have we have species suffering with habitat loss and also with habitat modifications. So the most concerning things are the agriculture and the pasture. So the man changes the land, and also the diversity suffers because they don't have any place to go. right? And that's why I think we should try to understand how taxa diversificate in this habitat. Then we can make conclusions about the past and how the forest worked to establish these high levels of diversity and endemism. So if we think about literature, of course, there's lots of process being discussed about the main responsible factors, the main active factors acting in promoting diversification. So we can mention, for example, the physical barriers, the geographic barriers that are established by rivers, for example. So we are not talking about these huge rivers that we have in Amazon, but we also have different rivers that are crossing the coast of Brazil. And we also have tectonic activity that we know that might have established some barriers in the past. So this may cause isolation for some taxa, and then we might have different conditions of evolution for different taxa in these isolated places. And we also discussed that the top of the mountains in the Atlantic forest might be acting as a quaternary refugia during the glaciations we have in the past. So probably these are stable areas during the glaciations that we can keep higher diversity during the not suitable conditions for them. So this might explain why we have such a great diversity in these areas. And there's also recent discussion about the sea level fluctuation and of course the continental platform changes that might have influenced what we can, like the richness and the diversity we see nowadays. And yeah, if we think about the frog, like the frogs are small creatures. They are super connected to the microhabitat because they if they independ of the water, like those species that I told you before that are direct developers, they're always tied with high levels of moisture. So they are interested in keep their skin moisture and do the gas exchanges and control the temperature. So they are really interesting creatures for us to study because they can tell us a lot about the environments. So imagine we can have like super interesting data by studying their preference, their habitat preference, their reproductive modes, and all these things. So that's why we can think as the mountains as islands. So they are ideal areas for us to study things about biogeography and evolution. So if we think about the process that we know that explain the biogeography and like the diversity we see across the world, we can think about vicariance, which is when we have taxa being splitted by barriers, as I show you the rivers or the tectonic faults that we might have in the territory. But we can also consider extinction. We can also consider that the species are not stable in a place, so they can disperse, they can migrate. So there's different things that might explain what the distribution we're seeing in the land right now. But also we have to think that the mountains have a special have been a special place for this organism during the past, during their evolutionary history. And this is what kept me and kept my attention, and that's why I was looking to this kind of organisms. And if we think about the gradients I was talking about, especially the elevation one, we might think that there's not enough difference between lowlands and highlands in a small range, as I told you. But if we consider individual like micro habitat conditions such as the ones that amphibians need to pay attention in, we can think about the biotic and abiotic changes in this kind of environments. So top of the mountains hold colder temperatures and also higher UV incidence, less levels of oxygen. And all this might be a little bit challenging for the species who intend to live there. And also this drives biotic changes. So the drastic changes in abiotic conditions might drive drastic changes in habitat structure too. So you might think on quantity, quality, and diversity of predators, for example, and also prey, like the food is changing, right? The whole community is changing according to the abiotic changes. And so this drives very cool specializations in this organism. So I'm talking about amphibians because I'm really into them. So I know more about these organisms, but I know that birds and small mammals, for example, they have lots of local adaptations to this kind of environment. So one of them is the tolerance to the extreme temperature. So they can be diurnal species, for example, and they can control better the exposition to cold temperatures. They can be associated to specific local conditions, for example, moss. They can live in the moss because it's a warmer and probably a wetter place to be. And we can have, for example, adaptations related to this body size or to the body color to live in this not super suitable environments. And this is super cool. And I'm going to mention here, so you're probably not familiar with all these names, but I brought this because I want you to show you that there's more than 20 whole genus of frogs that are all endemic from the top of the mountains in the Atlantic forest. So yeah, we can think about, there's some of the genus that I placed here, like they are in white. There's a species living in widespread elevational range. And there's the other genus that I'm marked on orange. They are specifically endemic from high elevation mountains in the Atlantic forest. Yeah, so we have, among them, we have the genus Brachycephalus that I am studying for more than 10 years. So I'm gonna show you a little bit of the data I've been collecting and what they can tell us about the history of the forest. So this genus comprise 38 species. So this is commonly known as a pumpkin toadlet, as you can see because of the color. So they're really tiny animals, and we currently know 38 species from this genus, and most of them are recently discovered. We still have lots of populations that we are working on the taxonomic descriptions. And we have a huge variation on body color, so you can check this plate. We have completely dual-colored species. We have reddish. We have different colors showing, and this is attributed the orange color is attributed to the presence of analogs of tetrodotoxin, which is the same as the pufferfish toxin that we can see, and is kind of dangerous, right, for humans. So we know nothing about their toxins. We just know that there's 4 species that hold the toxins in their skin. So more studies are being developed. So we need to advance a little bit on this too. But they're all endemic to the forest, to the dense part of the forest. They are microendemic. So this means that they have they occur just in one or few localities across their distribution. So no more than 5 or let's say 10 kilometers of extension of their range. So they're considered microendemic. The majority of the species are restricted to high elevation. So we recently discovered one of the pumpkin toadlet species living at the sea level. So we thought we are getting closer to understanding them, but seems that we have lots of things to learn yet. And they have a reduced body size. So this called also my attention because of this huge association they might have with the habitat. So they are listed among the smallest vertebrate in the world. So we can look at this. This is not updated, of course, but we have as the third position one of species of Brachycephalus with impressive 8.7 millimeters in snout vent length. This means the whole length of the body of the frog. So they are super small, and we believe that they evolve through the process of miniaturization. So this means that they show different characters, different morphological characters that should be tied with this evolutionary history, evolutionary way to evolve. So some of them are the hyperossification of skin, skull, and skeleton. So if you look at this gradient, I put an arrow in the bottom of the figure, so you can go from one extreme from another. So you can see the head, the ball shape, the circular shape is the head, the skull of the frog, and you can see the spinal the spinal cord in there, right? The vertebrae. And we have just bones in this staining, and we can check that the gradient goes from non-hyperossified at all to super hyperossified. So the extreme condition seems that they have a helmet and a shield, like a bone shield in their back. So these things are not completely understand, like their functions, but they are novel features in the skeleton. They are being related to this process of miniaturization. So this is another cool thing about these frogs, is that recently some researchers were trying to find fluorescence in bones, and they do discover that the bones, the hyperossified bones in some species of Brachycephalus, are also shiny under UV light. So they might be using this for some kind of interspecific or interspecific communication that we don't know yet, but we are investigating this too. And we believe also that they are miniaturized because we can see lots of features such as digital reduction, loss of phalanges, loss of bones, and this hyperossification I showed you before. So considering the taxonomic history of this group of species, we have artificial groups that we can identify, and then we have lots of species that has been described as Brachycephalus. Then the name, I forgot to mention, brachy is small and refers to their small head, like related to their neck. So they're small species with a small head, and this is like the original species described for this genus was placed as a Brachycephalus as pumpkin toadlet, and then later species like dual-colored species were smaller and described as in another genus called Psilophrene. And this Psilophrene genus was later synonymized in 2002, yeah. Based on osteological features, they consider the two genus together, and they were all nested, and we have to deal with this morphological differences, right? So they are still very different species in terms of body size. So we can see smaller sizes in the dual-colored species, and they have different skills to jump. For example, the dual-colored species are generally called flea toads because they jump a lot. They jump more than the pumpkin toadlets. And also they are different in the geographic and the elevational distribution. So most of the species that are in the top of the mountains, that is the majority of the genus, are the pumpkin toadlets and not the flea toads. So we have been working lots of years in this phylogeny for the group. And then what we have for this paper we published in 2020, it was great that we could identify that really the flea toads are amongst the Brachycephalus, so they are all same genus. We confirmed this. And we have identified two major clades, which might be related to the conditions, the climatic and topographic conditions in the Atlantic forest. So we started to investigate. I identified the main clades with the arrows in the figure, and then we tried to understand the contribution of the ancient tectonic faults of the Guapiara lineament. And this means we couldn't see a river splitting these two clades of species. And then we were trying to get back in the history of how the mountains emerged from the landscape and if they did something to contribute to diversification of these clades. And then we use georeferenced records of Brachycephalus from literature, from scientific collections, and we assessed the clades from northern and southern Serra do Mar, which is the main mountain range that shapes the Atlantic forest in the coast. And then we established different scenarios, like we constrained the ancestral conditions of the past, like the most recent ancestors, to see if they were dual-colored or aposematic, they were small, or if they were big, or, you know, we try to understand who were the most recent condition, who were the conditions of the most recent ancestor that can lead to diversification. So we perform lots of analysis, and we arrive in a conclusion that so the probably most ideal place for an ancestor will be at the city of Rio de Janeiro. So this is probably in this place we place the blue line in the map. And there's different mountain ranges we can see in these different colors in the map in the left. So we can see that there's a scheme of being dispersing through. So we thought that there was a parallel diversification between North Atlantic forest above the blue line and Southern Atlantic forest below the blue line, which correspond to the two main clades we have been found with molecular data. And yes, so these graphs are showing our analysis. I'm not going to explain that in deep for you, but we discovered really cool results. Like, for example, the errors in this analysis show the most probable direction for the evolutionary changes. So the north was probably the most recent ancestor were leaving, are migrating to the south, and the most recent ancestor are more probably restricted to highlands and were then migrating to widespread elevation. So we intend to trace all the history of these amphibians by reconstructing these ancestral states. And then we saw that there's a scenario of body shifts in the genus. So also here you can see the most thick arrow is like saying for us that the median species are most of the time turning to be small in the evolutionary history. So we can try to see if the ideas we have about miniaturization are correlated with these changes we are observing in their morphology, such as the hyperossification or the aposematism. So aposematism is related to the orange and bright color that some of the researchers are trying to associate to a way to avoid predation in this genus. So we don't know yet. We have no sufficient information to affirm that, to say it's true, but we believe that. And then we try to do some analysis to put these scenarios of coevolution together to see what happened. And we can see parallel evolution of larger body size between northern and southern Atlantic forest. So we expected to have larger body size in northern Atlantic forest, which is true. And then we have also this body size shifts accompanied by increased hyperossification and aposematism. So the larger clades we can see in the northern Atlantic forest are here in this figure. They are in purple and yellow. So the smaller size are the green color in this figure. And then we can see in this plate here, where we have the ossification of bones, that the purple clade hold the most extreme hyperossified species, as I showed you before in that gradient. So we have this. So huge increasing in the body size is probably holding their capacity to have a more hyperossified skeleton. And this is super cool. So we tested also if the body's color is related to their body size, and if the hyperossification is related to their body size also. So we checked that. And in these figures, we can see if you were looking to a large species of Brachycephalus, they probably will be aposematic instead of being dual-colored. And if you're looking to small Brachycephalus, they will probably be non-hyperossified. This kind of analysis we're doing by tracing the ancestral characters in their phylogeny. So we found no significant relationship of coevolution of these traits with elevation or altitude, which was good. Yeah, and this, like, we have lots of implications of the miniaturization to the evolution of different morphological characters. So we might think about their response to predation, or their response to the dry environment, dry conditions that we have in the top of the mountains. So if we check, this figure is a clear and double stained species. So we have the red parts, the pink parts are related to their bones, and the blue parts are cartilage, or sometimes are just mass. So the important here is that we can see they have osteoderms, right? So osteoderms are hyperossified warts that they have in the skin. And we believe that this might help them to work with the thermoregulation and also with the loss of water that they might have in environments that are drier, such as the top of the mountains. And also our results gave us an idea of, like, if they are getting bigger and they are like holding aposematic color, holding toxins, they might be more exposed to predators in these bigger sizes. So this might explain why they are becoming aposematic. And there's lots of things we can do still, like I'm still interested in knowing more about this species. So we are now interested in interspecific variations. So we were working in new species description. We have working in nomenclatural changes, because there's some taxonomic problems occurring in this genus that we might have to deal in the future. And we want to see also if there's some intraspecific variation, because these colors we might expect in different habitats might say something about them, right? We don't know what they want to say with this kind of colors, but we know that in different mountains, for example, we can have from a same species, we believe they belong to the same species, we can see different patterns of body color. And they might be related to, for example, ecological constraints that they might have in distinct habitats. And this is just to show you a really cool part of the process. So when we were studying their bones, we did this 3D models. So you can have, this is the Brachycephalus ibitinga, the last species I described. And we have the CT scan. Here you can see their bones by the ventral surface. You can see that they're really compact skeleton compared to the other anurans. And this is really cool. This is helping us a lot to understand like the fusion of bones and how they behave, how they use this these adaptations to, like how the diversity is related to these adaptations too. And then we advanced it a little bit. This year we have a really cool paper with phylotranscriptomic data. So we confirmed our past results that we have ancestral condition living in like as a dual frog, as a dual colored frog, and living mainly in the widespread distribution. So we confirmed our previous expectations. And then we also found really cool, interesting results in the mitochondria. So another thing that we show they differ from the other anurans in their genomes. So there's lots of things we can think about this genus. And the main message I want to show you was just like that we can look specifically for one taxon and then discover lots of cool and interesting things about where they live. So they are telling lots of things about how the mountains behaved in the past and how the communities, not even just of frogs, how the neotropical communities evolve and how they were linked to the past changes we have in their habitats. So yeah, we were trying to investigate other things such as the smaller climatic fluctuations we might have in tropical mountains compared to temperate or subtropical conditions. So they might have a restricted environmental niche because they are super small species, and we believe they do have lower dispersal skills. So we intend to test at some point when we have more data available if the mountains where they live are experiencing differences in the levels of diversification in this genus. And here is just to show you some of our efforts that we are continuously doing. So I have colleagues here. While I'm here in Canada, I have colleagues there in Brazil working, and we were trying to know more about their natural populations, where they live, what they feed for. I don't know, we are interested in knowing more to know if we can get more knowledge on them. So here are my collaborators. So my main supervisor on all this research has been Célio Haddad from the São Paulo State University. And also I have been working with Kelly Zamudio in the University of Cornell. Now she's at Texas. And we have collaborators across Brazil. We have Miguel Vences, the collaborator in Germany. We have now people working with me here in Canada. So I'm trying to share some experiences and get some cool advices from people working with biomechanics also. So we have lots of experience to share. And I'm super happy to share these results with you. And if you have any questions, or if you want just to share your impression about the frogs, so I'm free, I'm open to questions. And thank you again for hosting me.